cause them to be eaten. Now the hard part starts. So we got a finding, but it's a mouse antibody. And if I were to put it into any of us, we'd make an antibody against the mouse antibody and have serum sickness. That's what horse serum used to cause. So we've genetically engineered the mouse antibody and replaced all of the mouse DNA with human DNA. Now we got one that'll work. But we got to take it to clinical trials. So, well, we get maybe 50 to 100 patients a year in the leukemia service at Stanford, all with different insurance companies, as I said. We applied for and got money from a stem cell agency in California called the California Institute of Regenerative Medicine, which was put in place because of the ban on stem cell research. That was federal. So this was what you would call a states' rights issue, if you want to be political about it, which we were. And so we applied for, and with an expert review panel, got $20 million to take this through the phase 1 trial. So we don't have to start a company. We don't have to please venture capitalists. We don't have to look over our shoulders that a big company is going to buy us and shut it down for their reasons. And at the same time, the Acute Myelogenous Leukemia Consortium in the United Kingdom, which, by the way, has a National Health Service, and they promised us 50 to 100 patients every week. So they got a grant from their agency, $10 million. We got 20 for ours, and we're working together to make enough of the antibody in a safe way that it won't cause toxicity, won't kill a monkey in the one animal trial we have to do, and we take it to clinical trial. When we applied for the money, we had acute myelogenous leukemia alone, but since then, we have all of these. So, if you got 5 minutes, I'll tell you another short story, but otherwise I could stop here, because I think there's a lot of lessons that you've learned. If you take stem cell thinking, you apply it to tissues, you have the basis of regenerative medicine. If you apply it to cancers, you have a whole new way of looking at cancers and looking for weaknesses in the cancer. We were lucky to find this one. Nobody's leaving? So I'll just mention the controversial subject so you know what it is. As I said, when a sperm and egg get together, they go through about 8 or 9 cell divisions to make this. The blastocyst outside will implant in the uterus, inside will give rise to these tissue stem cells. So that, it was found, could be put in a test tube. And although they don't self-renew in the body, because every one of these become committed to this fate, one of those fates, in the test tube there's a chemical way to keep them at that early stage development. So to do that, people went to in vitro fertilization clinics where women and men who couldn't have babies the regular way had a sperm and egg put together in a test tube. And then they usually made so many of those blastocysts, which if implanted in the uterus could make children, that they had excess. And at the University of Wisconsin they said, you have the right to go ahead and try to make a stem cell line because it's so important, which they did. And that led to the controversy, which I'm not going to have to say too much about. So the stem cells that have all of those pluripotencies exist normally in the pre-implantation stage, and they're gone after implantation in the uterus. As culture cells, they can give rise to all body cell types, and we can expand them infinitely. So we know we can get enough cells. So they have to go through the adult stem cell, brain stem cell for brain, blood stem cell for blood, muscle for muscle, cardiac for cardiac, before they give rise to it. So the changes that they go through turn out, when you study them in the dish, are the changes that the cells go through to become tissue stem cells. So we have a chance to get authentic tissue stem cells. So there's a couple ways that you'd like to be able to work with those tissue stem cells. With the embryonic stem cells we have now, they represent the people who can afford to go to an in vitro fertilization clinic. There's, of course, a bias in the ethnic groups that are there. A significant population in Montana, I assure you, is not represented in the whole set that we want to use for learning and understanding and treating disease. So there's got to be a way that you increase the diversity of those cells if you're going to go through with what you say, that is to advance medical science to treat all patients. Then we have to learn, when they go through that transition to make the tissue stem cell, whether the tissue stem cell they make really works in a transplantation mode, and we still are working on that. And of course, once you have that, because of what I told you almost an hour ago, if you co-transplant an insulin-producing stem cell and a blood-forming stem cell from the same donor, here a cell line, you induce tolerance and stop the disease with the blood-forming stem cell, and now the tolerated graft is the insulin-producing, or the cardiac stem cell, or the muscle stem cell. So it's a new era that could come from this. Instead of remembering to take drugs every day, at some point, 5, 10, 15 years from now, we expect that from embryonic stem cell research, including much of which was banned, one could hope eventually to get regenerative therapies. Well, I'm going to just skip that and end by saying that there are ways now to get stem cells made of that type from adults. One of the ways, which I was going to show you, is by putting the nucleus of a cell in my skin into an egg that wasn't used in the in vitro fertilization clinic, shock it, and it'll go through 8 cell divisions. It can't make a human being because there's too many genes to fix up to make a human being. But it can make a cell line that, when you grow it, can make tissue stem cells. So that would have my genes. So I can tell you it would get asthma if you ran in the cold, and other stuff like that, and other stuff. Well, it sounds amusing and interesting, but what if we could take that from somebody with Lou Gehrig's disease, which has heritable foundation? And through the Genome Project, we know the 5 or 6 genes that could combine to make the disease, but we have no idea which gene is responsible for the gradual loss of motor neurons from here, that innervate from here going up to here, and ends ultimately and always in the death of the patient. But if we have a cell line from a patient with Lou Gehrig's disease, that we can put the brain stem cells into the brain of the immune deficient mouse, the muscle stem cells into the muscle, then we could look at the disease as it develops at that stage in the context of the body. And you could go back to the cell line and fix one gene at a time, because that we know how to do in a tissue culture. We can replace a bad gene with a good gene now pretty easily. So that you can say, okay, I found out it causes the disease, and if you fix gene 1 of 5, it didn't help. But gene 3, that really prevented the final development of the disease. And then you could even tell the pharmaceutical companies, well, the product of gene 3 is your target. Or the cell therapists, this is the gene you better make sure isn't the disease type, but is a normal type, where you're going to try to put brain stem cells into the brain to fix the disease. So it's another huge revolution that could come about. And that's all, folks.
We're happy to take questions, and there are members of our scientific research staff who have microphones, so if you just raise your hand and pass it to the
So, here's a question. Spinal cord injury,
If you can repeat
What if
this.
that injury is 3 or 4 years old?
So in spinal cord injury, the contusion type, the one that we studied, if you put the stem cells, the day of the injury, the stem cells get killed by all the toxic products of the injury. If you wait 9 days, it works. If you wait 30 days, it works even better. If you wait 60 days, which is the longest we've gone, it works even better. So it will really depend on whether the neurons that were denuded of the insulator are still alive and their processes, their axons, are still going by. We'll actually do the first clinical trial with fairly long time after the injury, and if that works, it would be marvelous because you know lots of people who that would help.
You were talking about the juvenile diabetes and possibilities there. Would that work the same with the neonatal diabetes?
So neonatal? Well, sometimes neonatal diabetes, which goes away, is because the mother has diabetes. Is that what you're talking about?
I can't remember the name of the gene that when they did the DNA testing. I can't remember the name of the mutated gene. But our son has the neonatal diabetes, but he's had it for 40 years. So he is too old for it to cure in the early years. And so I was just wondering if the stem cell...
But he takes insulin.
Yes. And now the glyburide too.
Yeah, well,
But I wonder if
there's a chance. There's a chance, but I won't go out on a limb to say exactly that it would happen for him. But remember, we got to go through a whole bunch of stuff to show that the kinds of stem cell therapies can be given to somebody, let's say it's a kid, without killing the kid. Because you already got insulin, which, although it doesn't work perfectly, and it's hard going through teenagehood, just thinking insulin, sugar all the time and not all the rest. But once we get past the toxicity and can transplant them, then of course we're hoping that it'll work.
There's one. If you could pass the microphone.
My question is, you give stem cells to mice which do not have an immune system. How do you give a stem cell to a person who has an intact immune system? I know in a leukemic patient you kill all leukemic cells, and a cancer patient you kill all the cancer cells. But let's say in a regenerative process, how do you keep the host versus graft reaction from attacking the stem cells, and do the stem cells not have a genetic identity to attack?
So what we've done so far is to give a dose of chemotherapy or radiation first, as you do in a bone marrow transplant. Then we found that if we give antibodies that remove T cells and natural killer cells, the ones that would reject the graft, we can lower the dose from a lethal dose to a sublethal dose. But we still need to make room for the new stem cells coming in, and that's what the radiation is doing, killing off the stem cells. So we recently found an antibody that selectively depletes stem cells. And we found it to deplete mouse stem cells, and we just found one that depletes human blood-forming stem cells, and maybe the cells that make freckles, melanocytes. But you could live with that. So we're going to see whether the combination of an antibody to T cells, an antibody to NK cells, and an antibody to stem cells collectively will work. We've just formed an arrangement with the group that has that antibody against human stem cells to see if it will work. That's still in what you'd call the preclinical testing stage. We could do it if somebody has a fatal outcome coming up where the irradiation would be no worse than what's going to happen. We could do that probably tomorrow, an allogeneic transplant. But we'd really like to work out the rest of the technique.
The stem cell you found her to start with, was that the blood-producing stem cell?
Right, so the first one we found was the mouse blood-forming stem cell, and using what we learned in the mouse, it only took us 3 years to get the human.
blood producing stem
It was the blood-producing stem cell.
cells that you've gone on to find.
Then we went on to find the brain-forming, then the muscle-forming. We just found the bone and cartilage-forming stem cell, and we're actually getting close for a heart stem cell, but that's a little tougher.
As you challenge the existing system, how many death threats have you received in your travels?
Well, I actually did get one pretty much early on. And I thought it was a joke. And I mentioned it to a guy who was the principal financer of the company I formed, and he knew somebody in President Clinton's security office who said, it's not a joke. So they called the FBI and they talked to the lady who wanted to kill me. And she didn't want to kill me anymore. And I don't know what happened.
So the Batten's disease, is that similar to Alzheimer's or dementia?
Batten disease is not the same as Alzheimer's. Batten disease affects children, but it belongs to a group of disease like Gaucher's, Tay-Sachs, Niemann-Pick, Sanoff disease, which are all failures of enzymes in the garbage dump of cells. Alzheimer's, though, is a really interesting disease because anybody you know who has that or other senile dementias loses the ability to learn or remember what they just learned first. And then they start losing other things. So I suspect, although I don't know, that the principal early effects are due to the loss of brain stem cells. Now, George Carlson has developed a mouse model of both mad cow disease, human prion disease, and Alzheimer's, and when we transplanted human brain stem cells into his Alzheimer model, you correct me where I say it wrong, that was in an immunodeficient mouse, even though it had the plaques that are characteristic of the developing disease but not the fully developed disease, the human neurons went right through the plaques and did fine. So we think there's something to learn there about Alzheimer's, and maybe it's cell replacement, maybe not. Was that right? Okay.
You mentioned the importance of diversity in your experiments. How important is that really? And do you have a politically acceptable or feasible way of actually doing that?
There is a politically acceptable that I went by, but we don't know if it's an accurate way to make stem cells from you that recapitulate your disease, because you have to use 4 cancer genes. And unless you get rid of the cancer genes, you just won't know. But let me put it this way. We had a way in 2002 where the experiment in mice showed if you take the nucleus from a mouse that's got immune deficiency, put it in the egg, make a cell line from that, you recapitulate a whole immune system in a mouse, it had exactly the same genetic defect. So we knew it could work, and then you had to find a way to do it probably first in primates because you don't have enough human eggs to do that experiment. But we had the way going, but that's what was banned. And so when I would argue this case, they said, well, you scientists are smart. Maybe, 5 years, 10 years, you're going to find another way, and maybe we do. Well, what if you were a family member, you got this window of time that you could be treated? How are you going to feel about somebody who said it was morally unacceptable to this segment of the political population of the United States? It was morally unacceptable to the president, that's why he made the ban. Well, I think he'd be pissed. And those parents of diabetic children were really angry, and that's why we have that states' rights issue, Proposition 71 in California, because they didn't want to accept a political judgment that prevented a line of research that might help people.
Dr. Weissman, we have a son that had a C7 spinal cord injury that was not severed and has gone to Germany for stem cell treatment, and he went for adult stem cell. And I don't know what your institute is working on towards that, but I'd like to hear what we could do over here.
So I don't know which German clinic he went to.
Excel.
I'm not going to say anything else. I don't think they have the no, I know they don't have the brain stem cell.
Yes, this was, I think it was
Mesenchymal.
bone marrow from the hip.
Right. Bone marrow, raised mesenchymal cells. No evidence whatsoever that they can turn into the insulating fibers. No evidence. This is exactly what you're worried about. That's why we're trying to push the brain stem cells that were proven to do it before we decided to take it to clinical trials.
Thank you.
So it's popular to say that the human brain exhibits neoteny, that compared to other primates, our brains retain juvenile features for a longer time. So if we're lucky, we can stay curious and creative a whole life, something like, say, a chimpanzee who gets old and boring. I was wondering if you think there are differences in human neural stem cell biology that contribute to this our ability to retain neuroplasticity into adulthood.
So this is a tricky question. So here's what I think. At the level of each neuron, I don't think there's a difference between mouse or human. The human brain stem cells put into the mouse brain didn't restore mouse brain functions. They stopped the disease, even when they were only at the highest level 1% of all the cells. Now, none of the mice came and asked us for better living conditions and many to come every night instead of every other night. So we have no evidence that a behavioral change when you replace one choice of a neuron with another choice. Now, if we were to take a mouse that during development had no neurons that could develop genetically, obviously a lethal, and then put into them human brain stem cells and watched what kind of brain they made, then we could approach the issue of, are there cell intrinsic species difference that makes human neurons growing up in the context of a mouse brain, in the size of a mouse brain, that would make a human screaming to get out, to get at what you're getting to. And I proposed that to the Stanford bioethics community, and I said, by the way, we're now getting to the point that we can get neurons, neural stem cells, brain stem cells, made from adult skin cells, from humans that have those set of diseases, plus schizophrenia, including its heritable form, plus bipolar disorder, including its heritable form. So knowing that we can put the disease cells into a mouse where they'll make up most of the neurons. You tell me if I should go ahead with the experiment. You tell me if there's something inherently wrong in trying to find that out. And don't tell me about Franz Kafka and metamorphosis. Don't tell me what you learned from fiction or what you invent by your imagination, because lives are going to be at stake to answer this question. And they came back and they said, do the experiment. It only took them 5 years. But then that's the pace of the ethics community. So, do I sound a little frustrated at times? No.
All right, it's just about 9 o'clock and time to stop. I'd like to thank everybody for coming, but most of all, I'd like to thank Irving Weissman for a wonderful evening.
And I'd like to thank all my friends and family who came, but also to remind you that this was a seminar put on by a research group that does biomedical research that will be translated, in the auditorium of a hospital that needs desperately to see these translations. And I hope everybody here helps us, helps the McLaughlin and Benefice find a way to work together so it moves even faster here in Great Falls.